Good evening, everyone. Welcome. It's great to see a nice turnout this afternoon, this evening. It's beautiful May evening. So we are excited to talk a bit about advancing drug discovery through innovation. And specifically tonight, we are going to have a conversation with Dr. Hermann Haller, who is the president of the MDI Biological Laboratory, and Jim Strickland, who is the senior director of research partners and translation at the Roux Institute at Northeastern University. So we're very excited to have them with us this evening to talk a bit about a relatively new initiative here at the MDI Bio Lab. And we want to try to put that into context with for you tonight. So talk a little bit about why we're doing this and specifically about some of the research projects that we are currently embarking upon as part of this initiative and what that means in terms of our ability to kind of move discovery from that sort of basic research element through the discovery pipeline to more of a translational application, which is more in line with developing therapies that can then be utilized to improve human health. So with that, I want to again just welcome Dr. Haller and Jim Strickland to our Science Cafe stage this evening and engage them in a conversation about this new initiative that we call MDI Bioscience. And Jim, I'm going to just jump in with you this evening and just ask if you can kind of help us understand the current landscape when we talk about drug discovery. What does that look like? And I mean, obviously there are some challenges there. So where are the opportunities to maybe speed up that process or make it a bit more efficient? Can you just help us understand that?
Sure. Thank you, Jerry, and thank you for the opportunity to be here this evening. I really appreciate it. And, I think when we think about pharmaceutical discovery, it's a long and laborious process that encompasses, billions of dollars and upwards of 12 to 15 years to get a drug to market. So there are many opportunities for finding efficiencies in that process. Certainly what has evolved is sort of a regulatory framework for drug discovery has evolved over a period of time. And, it really follows a very prescribed pathway through, discovery work to preclinical work where you're looking at model systems, then into human clinical studies, and then on into even after a drug is approved, there's post-market studies to see whether or not the clinical studies really did predict the real-world effect. So there's a lot of opportunities for efficiency there. And one of the things that excites me the most is, looking at that discovery phase of drug development and creating opportunities to use new model systems like MDI-PL has been championing, as well as, computational methods and other new technologies like AI that can really help bring technologies to market more quickly and with greater predictive efficacy. So those are the things that really get me excited, and it's one of the reasons that I'm so excited about the work that MDI-BL is doing.
That's great. So Dr. Haller, you have long been a proponent of these alternative animal models or really taking a more comparative approach to how we do our work, particularly here at the MDI Bio Lab. And I'm just wondering if you in your position as a physician scientist, can talk a little bit about why using these models, these what we call non-mammalian models, might actually help in achieving some of the goals that Jim was talking about, how to kind of move that process forward in a more efficient way. Can you tell us a little bit about that?
Yeah, I can, and I like to try to, and it's one of my favorite subjects. You can imagine one of the reasons I'm at MDI-BL because of this biodiversity, and it started early on. Some of you may remember that we were doing marine animals, so this is where biodiversity comes from. And you asked me the question, why are these animal models so important? So I'm a medical doctor. I'm interested in disease, I'm interested in diagnosing diseases, and I'm interested in treating diseases. So as a medical doctor, when I look at a patient, I first see the patient. But then in order to make a diagnosis, in order to define what's actually wrong, you need to understand pathways, you understand organ function and what's wrong. Easy example, for instance, in the kidney, when you have kidney disease, you excrete more proteins in the urine. That's one of the hallmarks. This is how we diagnose disease. And this is also then a therapeutic strategy, how to ameliorate the condition, that we reduce proteinuria. So once I have understood this, then I'm looking for model systems where I can mimic this pathway. And I don't need an animal model which is human. I need an animal model where the pathway is actually exactly modeled. And this is why our animal models are so important. So for instance, in the zebrafish, we have kidney disease, which really reflects what's going on in patients on the genetic level, on the molecular level, on the proteinuria level. So I'm not only the expert detecting with using a dipstick protein in your urine, I can actually detect protein in the urine of a tiny zebrafish.
Mm-hmm.
And this is what we have developed over the years. So we have several systems where we mimic disease, inflammation. We'll come to this in a moment. Kidney disease, we can do eye disease and mortal human diseases. So in a nutshell, we are like a hospital with fish and other animal patients, and then we can actually induce the malfunctioning of the organ. And then we have a wonderful model, a disease model, where, and Jim was referring to this, we go into this difficult process, finding new therapeutic agents, and we can shorten this. I'm a great friend of shortcuts. And here is exactly a shortcut where we use one animal model and then we get vital information which saves us months and sometimes years if you're only stuck with one animal model. And this is today very often the mouse, and eventually we'll go to the mouse, but in these early stages, our animal models are the best.
Fascinating. So Jim, getting back to, your comments around the current process, clearly this is an expensive endeavor, right? So we were talking earlier and you used the phrase de-risking. And I'm just wondering if you can talk a little bit about how this, how using these animal models on the early stage, in the early stage of drug discovery, can help eliminate or minimize some of the risk.
Sure. So I think when we think about the first stages of drug discovery, a lot of it is, we do a lot of in vitro assays. We look a lot at how cells respond in basically a dish, and whether or not there's a signal of efficacy based on how a cell responds to a compound or a molecule. What's missing is then we do these huge jumps into mammalian species, and there tends to be a big expense associated with that jump, and also a long lead time to get the compound spun up enough and enough quantity that you can do those types of tests. There's a need for a middle ground there where you can use non-mammalian systems like zebrafish or turquoise killifish or even C. elegans, where you have a whole organism with different organ systems all functioning in unison, and you have that sort of unique balance of biology working in its intended way. And you can actually study that organism over a period of time. In cells, you really have a very finite amount of time where you're sort of keeping them viable. in an organism, you can really do a study over a much longer period of time and see how an effect might elucidate itself over time. Or if you're seeing a negative effect, such as toxicity, then you can also see that in an organ system earlier than you would if you were to move into a mammalian system like mice or other animals. So it really gives you that early window into a whole systems biology, which is a very valuable tool in drug discovery.
Yeah, that's great. And so for our audience, if you all have questions as we are, having this conversation, please just feel free to raise your hand. You can use the icon to raise your hand, or you can take yourself off camera and physically raise your hand if you'd like. That's fine. We'd love to have this be a dynamic conversation. So, Dr. Haller, in thinking about that and why, zebrafish are particularly well suited for this kind of work, can you describe for us, some folks may not realize that zebrafish, when they lay their eggs, those eggs are actually part of what we are using in these tests. And so they're transparent. There are a lot of qualities about those eggs that make them very suitable for this kind of work, right? As opposed to a mammal where you have in utero, develop.
To be honest, it's not only it's the embryo. So they have the eggs and they're transparent, and then the embryo grows, and these fish embryos, they are tiny, they're small. They are, at least in some countries, not even considered to be animals, but they have all, not only the genes, but they have the organ function. It's always one of the most fascinating things for students when we train them, that when we cut the kidney of a young embryonic zebrafish, the kidney looks under the microscope like ours. So there are glomeruli, there are blood vessels, and so you can imagine that these are wonderful models because we have it, as I mentioned before. And then we can manipulate these animals. We can, especially since they are transparent, we can express some marker genes in there. So these are using advanced methods like CRISPR-Cas. We have then fluorescent fish where the function can be visualized, so we can see it under the microscope. So we do two things, actually. On the one hand, we have fish, and they excrete green fluorescent protein when they have kidney disease. So we can see this, and we can measure it, and we can measure the effect. At the same time, we are using the whole small fish to do gene expression profiling. So with the novel methods, with bioinformatics, we have now the tools that we look at organ function, but at the same time, we see the changes in all the different systems. And you can understand how powerful this is when you have your animal model. The genes are very comparable to ours. So the information we are getting using these novel ways, even artificial intelligence, to provide us insight what a compound, and this is what we are interested in, whether this compound heals the defect and what other effects the compound has on the genetic pathways inside the fish.
So we've kind of set the stage now for, some of the work that we'll be doing or are doing in MDI Bioscience. Talk to me for a minute about partnerships, because I think, folks who have been following us along as we've developed MDI Bioscience know that we have established a robust partnership with the Bigelow Laboratory, and we are doing some screening of algal compounds there. Can you talk just a little bit about that, Dr. Haller, and why this is a particularly great example of how to utilize these models to develop new therapies?
Yeah, I'd love to. This is a marriage in heaven. Actually, it's a marriage in Maine, but this brings together two institutions which are rather unique. So we have now described our animal models and what we can do with these animal models. And the Bigelow Lab has a collection of a unique collection of algae. They are one of the national centers for algae collection, and they have started to look into compounds from these algae. And then Jim Strickland was giving a talk there and explaining what we are doing at MDI-BL. And then their president said, "What? You have all these animal models we could use, and we are in the same state?" And the next week, she actually had to testify in Augusta. And she told everybody that MDI-BL came for the rescue for their wonderful algae project. And yes, it's exactly like this. So to be blunt, they are grinding up their algae. We have extracts and we have regular meetings, how they do this. They are very experienced with that. And then we get either extracts or already diluted samples. And then the question is, can we test this and what animals models do we have? Jerry, am I allowed to show a slide?
Sure, absolutely.
Okay. Then I can. Can you see that?
Yes.
But not in a perfect way, but now I think it should fill your whole screen. So
Yes.
that's the MDI-BL Bigelow Bioprospecting Program. Not all people like the word bioprospecting because it's drug development. So bioprospecting is more like we are out there with our wellies on and we are looking what else we are doing. It's not like this. It's highly specific. We are in the lab. And you can see the group working on that. So it's Jim Strickland started it. We are working with Yanni Becker, who is a graduate student in my lab. Jim Kaufman is involved because he developed one of the earliest models, and myself. But you see now the Bigelow Laboratory for Ocean Sciences, the purification process from 1,500 different algae strains. And I can ask you or the audience directly the question, who actually believes that algae are positive? I think everybody agrees that algae are positive. They may be slimy, but the ingredients are good. I mean, either you eat it or you use it for skin or for shampoo, or, I mean, there are lots of substances in algae, and we are trying to find out what's happening. So these are the animal models we have at the moment. So we have a zebrafish model of inflammation. We induce inflammation by exposing them to copper sulfate. Our readout is a fluorescent NF-kappa B, which is the marker molecule for inflammation. And this means then that we can use this to test for anti-inflammatory compound. We have already shown that in our control experiment. So we make the fish inflammatory, and then we treat them with ibuprofen, and the inflammation goes away. So that's a promising animal model to test now new ibuprofens. We have complement system inhibitors we want to test because complement system inhibitors are now all the rage for rare disease. We have a zebrafish model of acute renal failure, and we can see this actually because we have made a transgenic fish where the kidneys light up when they are exposed to gentamicin and other toxic drugs. We are working together with the University of Maine. They have a wonderful research group, Rob Wheeler, who is one of the world experts on infection in zebrafish. And we are working together with him. Actually, we have the animals now here. We look for candida infections because you all know that these are very important and difficult to treat infections. And then obviously with our expertise with Romain Madeleine, who is one of the world experts on eye disease in zebrafish, we look for retinopathy. And last not least, we have developed this African turquoise killifish model, which is a model of aging. And there we can use anti-aging compounds. And this is especially important because here we get the first data on whether an agent is useful to prevent the aging process. You can't do this in humans. You have to wait for 35 years before you see an effect. So we need animal models, especially something like this. And Terry, I have another slide, which I can show later on or right now, with one very nice example how this all works.
Sure. Show us now.
So this is another word we are using. This is drug repurposing. So some of you may have heard when we testified before the Appropriations Committee, the Appropriations Committee is all about money, and drug repurposing means we have drugs which are available for specific diseases, but they may work under other circumstances. And this is then, you can imagine, less costs, much less costs. On the other hand, not a lot of people are doing this out there in the pharmaceutical world because they are not patent protected. So this is more difficult than to find new targets. And this is what we're doing. Here you see on the left-hand side, the African turquoise killifish. So meet these wonderful fish. The females are smaller, the males are bigger. And then you can follow them through week 4, week 8, week 12, week 18, and you can see that they are getting more grayish, that they lose color, and you can see with one glance they're aging. Especially in the males, they are a little bit more bent. You can't see this here. So they have osteoporosis, they have vascular disease, they have heart disease, they have all signs within 24 weeks. So we were interested because we are interested in blood vessels, and we believe that the younger your blood vessels are, the better. So we looked into the microcirculation. You can see this in the middle panel. So we perfuse the kidneys, but we have done this in other organs as well, with labeled albumin, and you can see this wonderful meshwork of the small capillaries, which are not only in the kidney, but in all organs. And then when we zoom in, and you can see an enlarged picture of that, this is really a fascinating network. All these white things you're seeing there are tiny blood vessels where the blood is coming through, providing the tissue with oxygen and substances we need for survival. And then dramatically, and this is the first time somebody has done this in this model, when you look in old fish, you can see with one glance that there are much less blood vessels. So the capillaries actually retract, they become less. And when you zoom in, you can see this dramatically. Imagine these are old kidneys, and there is nothing wrong with them. They have just aged. And then we use a drug which is on the market, and I've been working on this in other models before. These are SGLT2 inhibitors. These are drugs which have been developed for diabetes, but now they're used in kidney disease, and they are also used in heart disease. And then we treated our animals relatively early on, from week 8, with these inhibitors. And then you see how dramatic the changes are for the microcirculation. We prevent almost completely the loss we see during the aging process. So this is very novel, but you can see the power of our models that for the first time we can test something. The drugs are on the market, so now we can do studies in humans. And then on the right-hand side, you see the power of gene expression profiling. So here you can see that we have used, just to make this readable, 6 inflammatory molecules: NF-kappa B, CRP, TNF-alpha, interleukin-6, and TLR7 and 21. And you can see the expression of these, and you can see when you're young, you have a very low expression. This is in lilac or in white of these inflammatory molecules. But when you age, the inflammation becomes more. And we have other researchers. Sam Bagg was here, and we published a very nice paper with him on inflammation. So inflammation and aging go together. And then on the right-hand side, you can see what happens to this inflammatory process when we treat with SGLT2 inhibitors. So no ibuprofen, no aspirin, but SGLT2, and we were able to really turn this back. So you can imagine we are very fascinated by this. On the one hand, we have a drug which may really have anti-aging properties. And the good message is the side effect profile of this drug is not negligible, but very small. But on the other hand, I'm now burning to find out what the mechanism is. What is the anti-aging mechanism? Our hypothesis is that we are basically reprogramming the cells to become young again. So in a nutshell, this shows you one example. And as you have seen with all the animal models, I've shown you this before, we have quite a few of them. So this is not only small, but together with the Bigelow project, this will be very important. And hopefully we can publish a lot of very interesting papers, get patents out of this, and influence the world of algae used for treatment of disease.
That's great. That's a great example. Thank you. So, Jim, I know there are a lot of other projects, as Dr. Haller outlined in that slide, that we're moving through this process. And I'm wondering if you can tell us just a little bit about Romain Menard's project. I'm forgetting the compound name at the moment, but that's to me another great example, sort of a different, type of application, but yet something that really has the potential to develop a new treatment.
That's right, Jerry, and it's really hard to follow those data, Professor Haller. Those really remarkable. But I think when we think about other projects coming out of MDIBL, ML233 is an existing compound that was being actually investigated for a completely different application when Romain Madeleine and Romain Menard discovered that when it was introduced into zebrafish, which are transparent, and you can see the pigmentation of a zebrafish quite easily. But when you introduce ML233, it depigmented a lot of the cells. So it really became a very interesting finding in its own right, and has led to a separate program for translating that discovery outside of the laboratory environment and into a more commercial posture. The interesting thing about that is we were able to patent it as a different use for the same molecule. It's because it's commercially available, we can buy it off the shelf and test it, which is terrific. And it's sort of another spin on a repurposing of an existing compound. But the project itself now is they've put in for a small NIH award, which they're waiting to hear back, but we're all hoping that they might be able to secure a little bit of funding so that they can move that project forward. The next step in their journey is to then try to use a skin on a chip model where we can actually look at human cells and how human cells actually react to ML233. Do we see that same effect on pigmentation? And it's really relevant for skin diseases where you have differential pigmentation rates, and it can be like vitiligo or diseases of the skin. It's really an important potential finding. So we are now sort of plotting how we can move this into the hands of an entrepreneur and hopefully out into a potential spin-out company that would allow the technology to raise some money to move it forward, which is a very exciting project, and it's been fun to be involved with. So
yeah, that's great. So I love kind of those two projects as a way to talk about sort of what this pipeline, what the whole discovery pipeline looks like, right? So we have two examples of what I would call sort of, discoveries that came out of basic research. The intent was not necessarily to identify a compound to treat skin disease, right? But it was one of those sort of, serendipitous discoveries that happened during the course of basic research. And Dr. Haller, with your project, I think, that also is a great example of this kind of asking big questions and trying to understand, using these models to understand whether or not that is, possible, or whether it's a true hypothesis. And so I want to just talk for a moment about the importance of both, right? The importance of basic research and how that can then lead to what we call translational research, or research that is potentially translatable for human patients. And Dr. Haller, I know you have a lot of thoughts about the importance of continuing, basic research and how that works. Tell me a little bit about how you see MDIBL maintaining both of those things, right? Maintaining a commitment to basic research, but also helping scientists move their work forward.
Yeah. Well, thank you very much, Sherry. This really is a very important question for me, and I'm very passionate about this, and I have very clear views about that because of my own background and where I come from. So I'm an MD PhD. I'm interested in disease. I'm interested in basic research. I'm interested in treatment. I'm interested in understanding this. And what we do with this concept of MDI Biosciences, we try to be very clear what is basic research. This is curiosity driven. This is trying to understand the world. This is fun. It's hard work, but it's not directly always into a project which will be translatable. Then the beauty is if you find something which is translatable, then it changes almost everything, because the translational research has then a different focus. It comes out of curiosity-driven basic research, but then it's very focused. Then we have to work to get a treatment strategy. Then we have to work to get a drug. Then all these questions about patenting really make it successful in the real world. And I've always been interested. I love working with pharmaceutical companies, and I like the curiosity-driven research, but we have to acknowledge that you need a different energy. It goes into different directions, and this is why we have these two. We have MDI Biosciences, and whenever our researchers like Roman has such a wonderful discovery, then we put it under the roof of MDI Biosciences and try to develop it and keep it protected, or better, the other way around, keep the basic research protected from these real world assaults where you think about, and you have to think about money, you have to think about how to translate this in the real world. And these different aspects are tightly connected, and this is the beauty of translational research. But in order to develop this and in order to protect our basic research and this, I don't want to call it playground of curiosity-driven research from the real-world application, this is why I feel so strongly that we need something like bioscience in order to define this is a project which goes in this direction, and basic research is where we do wonderful research and trying to find out what's actually happening. And allow me just one more example. Without curiosity-driven research, we wouldn't have CRISPR-Cas. Without curiosity-driven research, we wouldn't have the Human Genome Project, because you have to be interested in bacteria living in very hot springs And then you find out mechanisms which are important for these bacteria, and then you translate them eventually into something else. So we need both things. Both are fascinating, but when you do not have MDI Bioscience, and I didn't have this, so I made discoveries, I made patterns, and then I was like in Aristotle with this ass. I didn't know what to do. Did I want to go one way and become a very rich man developing these pharmaceuticals, or do I want to be a researcher and do curiosity-driven research? And when you don't make this very clear, you fail on both ends. I didn't fail completely, but no billion and no Nobel Prize. So you better make this distinction early on.
That's great. Well, I mean, I think that's it's probably not something people really think about all that often in terms of how research is structured and, how those projects are developed and how they get closer. They also probably don't think a lot about what different resources a scientist might need, when they sort of get to the point of needing to take their project forward into a more marketable approach. And I'm wondering, Jim, I know a lot of that is the work that you are doing at the Roux, right? To create these resources to help train folks to have a more entrepreneurial focus to their work. And I just wondered if you could talk a little bit about that. Where do you see, or how do you see these kinds of initiatives really helping scientists be able to do exactly that, to take that work forward to the next step, or even to hand it off to someone who might want to take it the next step?
Right. It's such a fun challenge to come across sort of a breakthrough moment in science and then be able to sort of ask three really fundamental but de-risking questions. Earlier you mentioned de-risking, and that's sort of what translational science really, sort of first and foremost asks, is it novel, right? Can what you have discovered, has somebody else discovered it first, or is it patentable? Second is sort of, can it be applied at scale? What are the key factors to actually extrapolating that discovery out to a marketable product in mass that would potentially prohibit that translation across the spectrum from single user to multi-user environment. And then the last question is, who would benefit, who would buy it, and who cares? And those questions are often the hardest questions to broach with a researcher, because of course, it's they've spent all this time and money. Everybody will want this, and as somebody who works with researchers and sort of entertaining these three areas of questioning, that's the one where oftentimes you have to really dispel a confirmation bias. It's not necessarily that it's a bad idea. It's just maybe it needs to be modified in a different way to make it more applicable to market, or maybe it needs to be an analog of that molecule so that it can be manufactured in a certain type of way that makes it cheaper. And so, I liken it to the Wright brothers, right? When the Wright brothers started flying, their airplane was an incredible discovery. It was amazing to see human flight for the first time, but they were the only ones that wanted to fly in that aircraft. And so, to translate that out to a broader audience, it needed landing gear, and it needed a windscreen and comfortable seats, and so those basic discoveries are the foundation which translational research sort of builds upon. But ultimately, it's a necessary step to building value from that basic discovery at a very broad level.
Mm-hmm. No, that makes total sense. So, in thinking about kind of all of these different elements that we have, I want to turn to the current sort of ecosystem in Maine for discovery science and drug discovery work. And I'm thinking a little bit about all of the different, pieces that are beginning to come together in Maine, I think, to create a more robust infrastructure for this kind of work. We've always had strong science, in Maine with a lot of our institutions, but really moving that forward and developing more of a R&D sector in Maine has been a relatively new enterprise. So Jim, I know you sit kind of in a great place to see how that is progressing. And I just wonder if you can talk a little bit about where you see the need, is something like what we're talking about tonight, how does this fit into that ecosystem? And then where do you see things going, just in terms of the state's development of this sector?
Thanks, Jerry. What a great question. I think, Maine is such a unique place. If you looked at the research organizations around Maine, it's really remarkable how many tremendous independent research organizations there are in Maine. And certainly great universities too, with University of Maine and Northeastern, but and many others. But it's going to take a community really to sort of build, particularly around drug discovery, an ecosystem that's capable of really generating new innovations in large scale. And so it's been fascinating to see how the state has come together around different initiatives and building a life science economy. Certainly, there are assets here between MDI BL and Jackson Laboratory and Bigelow Lab for Ocean Science, and that all sort of play together in a very nice way and sort of create opportunities for a drug discovery ecosystem to emerge. And if we play to those strengths, I think it becomes even more compelling as we think about, how genetic medicines and different types of modalities can actually, create a new pathway to different therapies and different treatments for patients of the future. But certainly, I think one of the things that we touched on earlier is how do we make it more efficient? And that's where I see, there are some real assets that Maine has in, particularly in genomics and use of model organisms, and also in computational methods where we can bring these assets together in a very unique way that really can add up in terms of time savings and cost savings in the drug discovery realm. And I think that's something to be excited about as a state.
That's great. And do you feel that there's an opportunity to train? We don't really train scientists to be entrepreneurs, right? We train them to be scientists. And so I'm just curious how you're finding that as you have discoveries that are coming, or that are being furthered toward application. Are you finding that Maine has that sort of infrastructure to take those discoveries to market?
I think we're getting there. And, one of my favorite questions that I get from scientists is the what's next question, because I do find that certainly researchers in Maine are asking that question about, I've discovered this, what next? How do I create impact out of my science, out of my research? And I think that's a really promising signal. And I think it's one that, we are starting to see more and more translational research come out of places like Bigelow, where they're doing some remarkable work, and MDI BL with MDI Bioscience. And so I think that we're building up this translational asset in the state and sort of working together to do that. And when that sort of capacity is really kicking on all cylinders, then I think that is really going to be combined with the research prowess in the state. I think that shows that we as a state can do unique things, and we can pull together and bring the capacity needed to generate a real life science ecosystem to the future of Maine, which I think is a really great signal. So
that's great. And there are a couple of initiatives or legislative initiatives right now before the state legislature, right, to support activities that would really, I think, help in some ways build this life sciences economy. So one, of course, is a bond issue that we've seen those kinds of things in the past and really seen the benefit of them. But there's also a new initiative, LD 1643, which is an act to establish a life science innovation center in Maine. And can you help us understand how that might help in what we're talking about, how this could sort of coalesce the life science sector in Maine a little bit?
Definitely. I just sort of described the team sport of Maine science, right? That we all need to work together and sort of be coordinating across organizations in order to fully generate impact and capacity. And the LD 1643 is a great example of how we can sort of convene a life science center that becomes sort of that organization and that pulls everybody together and says, hey, what direction are we going as a state? How can we increase the assets and resources available to sort of advance a strategic imperative as a state? And I think that would be really exciting to see that bill move forward and come to fruition, so that we can coordinate and really realize the potential that we all know is here, but we just want to mobilize it in a way that it hasn't been mobilized before.
Right. I've said for years, Maine, we have a Department of Tourism, we have, all of those things. Maine sort of needs a marketing arm for its life sciences sector, because I think a lot of people don't really see Maine as a place where there is a lot of life science activity. And that's part of our challenge, is to really change that perception. So for me, I think this is an exciting opportunity maybe to do a little bit more of that and to have folks see Maine as a place where innovation is happening. So it's really exciting. I want to take a minute and just open it up to the audience and see if folks have questions. I'm just, could be anything from sort of where Maine is now in terms of its life science sector, or it might be specific questions you have or curious about how the drug discovery process works. So if you have a question, just take yourself off camera, raise your hand. We'd be happy to try and answer that for you. In the meantime, while folks are thinking, Dr. Haller, what are you most excited about these projects in MDI Bioscience? Where do you see this headed? What do you hope the outcomes are for these projects?
Well, obviously, you have seen this anti-aging, and we are testing other drugs. And so this is a fascinating system. I also think that retinopathy is very promising and anti-inflammatory. But I'd like to mention something which I think is really very important. MDI-BL is contributing on two levels to what Jim was just mentioning, the state of affairs in Maine. And one is the quality of the science. The program we have at the moment, I think that's very important, is really on a very high international level. This is something we are proud of, and Maine can be proud of, but most importantly, we can use it for the training of young people. So when they are trained on that level, then they can stay in Maine or they can move somewhere else, but it provides a level of competitiveness which we didn't have earlier on. So we need more PR, but now we have actually with the institution, it's not only MDI BL, but we are very open. We are providing these resources to others, but also the other institutions in Maine. And I'm very proud what happened over the last couple of years in Maine, that we are up not only 1, 2, but 3, 4 notches from where we were before. The Roux is important, our development is important, the Jackson Lab is contributing, and to work together, we are starting now, this is another example for this educational process, which is important. We're starting a real research project with the Jackson Lab on Alzheimer's disease. And we're doing research together with them on other neuronal diseases. So all of a sudden, we are generating a level of international competitive science, which is so important for training young people.
That's great. Particularly in a time when it's not easy to make the decision to go into science, right? So these are tough times for science. So we need these kinds of initiatives to keep people engaged and excited about moving forward. That's great. All right. Well, if there are no questions, any last comments, Jim or Dr. Haller, for our audience to take away? Anything you want folks to know about this process that you're particularly excited about?
I mean, the good thing is that we were so convincing, but I'm really astonished that not everybody is asking, where can I get these anti-aging drugs?
I know, right? I was going to ask you that. I think I need to, exactly what age should we start taking these?
Jim was talking about a drug where you can actually change your complexion and you look much better afterwards. I mean, you know, we are, I think, with that research and patented research, we are addressing the central problems of this planet.
Right. When you did those studies, Dr. Haller, in the African turquoise killifish, did you see an extension of lifespan, or was that not the focus of that particular work? I'm just curious how much, if they lived a lot longer.
We didn't see an extension of lifespan. To be honest, we saw something, but our numbers were not big enough because these are small elements. So the traumatic changes we saw in the microcirculation, but to be honest, I'm more interested in the microcirculation than in lifespan, because lifespan can come from different things. But to be honest, if I would be able to keep my microcirculation the same way, I can hike Italy 4 times.
Excellent. All right. Well, thank you both so much. This was a fascinating conversation, and I think really bodes well for the future here in Maine. And we really have a wonderfully vibrant life science community. I love the way there are so many partnerships, and people are really, you know, looking for opportunities to kind of make the sum of the parts greater than make the whole greater than the sum of the parts, which is really exciting. And I think it's a great trajectory for the state. I do want to remind everyone that this is the final Science Cafe in our winter series. And so in June, we will be going back to in-person Science Cafes. And our first in-person Science Cafe will be on June 9th, and it'll be here, obviously, at MDI Bio. It will be offered hybrid, so we will have a Zoom option. But we will be bringing back our collaborating friends from the MDI Historical Society and the Landscape of Change Initiative. So that's always an exciting topic people are eager to engage with. So we'll bring those folks back and have an update on where they are with their sort of citizen science initiatives to look at the impact of climate change here on Mount Desert Island and sea level rise, and learn a bit more about how they're progressing with that work. So we encourage you all to join us in person if you're able. We'd love to see you, and hope you'll tune in for that next Science Cafe. And again, a huge thanks to Dr. Haller and Jim Strickland for joining us this evening to talk a little bit about drug discovery here at the MDI Bio Lab. And we look forward to seeing you again soon. So thank you all so much.
Thank you.